whose career we're all following with avid anticipation, and when we see you're a very famous person in the world one day. But I introduce you, Ethan. I don't need to say anything about him. We all love you and hope for your success all the way. We're coming there with you, and thank you for taking us on your journey. Yeah. We really appreciate it. Thank you.
Thank you so much, Valerie. It's so lovely to be back. Thank you so much for welcoming me back and being willing to spontaneously listen to me for no reason other than, I don't know, who knows? But I'm so happy to get to be here and to have an opportunity to thank you all for supporting me so enthusiastically over the past year before I went to Oxford, and now, I mean, I really missed giving U3A classes, but now I'm hopefully going to repay a whole year's worth of U3A classes while I was gone in just 1 hour. So that's my mission today. So welcome back to The Brain and Its Mysteries. I'm going to tell you a little bit about the year that I spent in Oxford doing my Masters. I'm going to tell you a little bit about the things that I've learned and hopefully pass on some of the coolest bits of neuroscience that I got to participate in. And then I'm going to tell you about kind of what this year has done for me and what I'm hoping to do next. So if that sounds good, I'm just going to jump right into it. So I want to tell you about my year in Oxford, also known as the best year of my life, in brackets so far, but we'll just focus on the best year of my life. There are so many things that I could tell you about this year. I could kind of, I think that probably the best place to start is with my college. So every student in Oxford has a college, and I have a video of me and my parents who came with me for the first week walking into the central square in Oxford for the first time and seeing the big library in the centre of Oxford called the Radcliffe Camera, and then right next to the Radcliffe Camera is my college, Brasenose College. So this was our reaction. That's my college. Remember that? Yeah. So I was in disbelief, right? In disbelief that after many months of not even knowing if I was going to be able to have funding to go to Oxford, it finally all came together and we were there, and it was actually happening. And the college that I was in, kind of for each student it becomes their home for the year. So Brasenose College is a beautiful college. I think it's the best college of all of the 40 or so colleges in Oxford. Completely not biased at all. It's right next to the Radcliffe Camera in the centre of Oxford. It has a beautiful chapel where I listen to my friends sing in the choir. It has a beautiful dining hall where I had lots of fancy dinners, and a library where I spent a few too many late nights and early mornings studying. There are so many things that I want to tell you about the year, and I could kind of group them into these different categories, but if I went one by one through these things and I told you about the whole year, I think you would miss out on some of the twists and turns and the cliffhangers and the surprises that I went through throughout this year. So instead of going one by one through these things, I'm going to follow the Oxford terms and go chronologically. So Oxford has 3 terms. They're called Michaelmas, Hilary, and Trinity. And they're called that because, actually I have no idea why they're called that, but we're just going to have to go with it. So the first term is Michaelmas. So that was from September 2024 to December 2024. So the last third of last year. I came to Oxford in Michaelmas. I settled in, I found my beautiful college, I found my room, I got my bike, essential in Oxford. I got my subfosk, which is the fancy academic dress that we have to wear for exams and for ceremonies. first formal dinner and checking out the local falafel shop. I started my course. I got my student card. I became a student. This is a photo of me in the Sherrington building. And the Sherrington building was really special to me because it was named after Charles Sherrington, who was a kind of a founding father of neurophysiology. He made some discoveries, Nobel Prize-winning discoveries actually, about the way that neurons communicate at the synapse. And he was a mentor to a young scientist named Howard Florey, who the Florey Institute was named after. So it felt like a cool piece of history to be tacking onto by going to the Sherrington building where Howard Florey trained. So this is us on our first day. I met my brilliant course mates. I think I told you all that I was excited to meet 20 other people who were just as nerdy about the brain as me. And I certainly got that. I met a whole bunch of brain nerds. But coming into this class, I felt super inspired and very excited, but I also felt a little bit out of place. My course mates did their undergrad degrees at Cambridge, Harvard, MIT. One of them is a fully qualified neurosurgeon, just decided to do this course for fun. There were 4 Rhodes Scholars in our course. 5 of them had guaranteed PhD offers, which I didn't have, so there were some fairly reasonable reasons for me to feel like a small fish in a big pond coming into this course. But I was super excited. This was me and a few of my course mates on matriculation day, which is the formal ceremony by which we officially become members of the university. So we all dressed up, went to the big Sheldonian Theatre in the middle of Oxford and paraded around and stayed in our fancy dress for the rest of the day. This is 2 of my neuroscience friends doing brain surgery on me on matriculation day. And then this is my friend Kate and I doing a handstand on the lawn. And that was extra special because matriculation day is one of the only days in the year that you're even allowed to be on the grass, because normally they want it absolutely manicured. So that was very special. So at this point, I think you can kind of see that I was falling in love with this place. And very quickly, I mean, even before coming to Oxford, I knew that I wanted to do a PhD. And very quickly, I knew that this was the perfect place to do my PhD. So in Oxford, they call a PhD a DPhil. So instead of a PhD standing for a Doctor of Philosophy, a DPhil stands for a Doctor of Philosophy. So it's the same thing, just Oxford likes to call it different things. But this was when my quest for the DPhil started, right at the start of my Masters in the first term in Michaelmas. Because I put my applications in for starting a PhD the following year. I applied to 2 different programs at Oxford and 1 at Cambridge. And I put these flowers there because they were a gift from my friends when I submitted my last applications. And it was around this time when I realised that getting funding to do my PhD at Oxford would be a long shot because of all of those reasons I mentioned. You know, I didn't go to a world-famous university for my undergrad. I didn't have a previous master's that I could use for my application. So I knew it would be a long shot, but what I was told by some more senior students was that if you get a distinction in your master's and then you reapply the following year, then you're much more likely to get funding. So I set myself that goal of getting a distinction in my master's. There was lots more for me to do in my course. I actually had to get to work, so I listened to some lectures. This actually is a lecture that is especially relevant to us today because Guido Miesenburg, who was this lecturer, he was one of the people involved in the discovery of a technology called optogenetics, which I did some research on in my first thesis project, and I'll tell you a little bit more about later. I did lots of studying with my friends in beautiful libraries, and here's me getting shocked by a transcranial magnetic stimulation machine by my course mate. It's not actually turned on. I also did some stuff outside of neuroscience, can you believe it? I tried trampolining. I had been a springboard diver when I was in high school, and Oxford didn't have a diving team, so I thought, what's next best? Well, it's probably trampolining, so I did lots of flips, we got to meet a beautiful team, and then I went to my first competition, which was in Bath, so I got to travel around a bit around England, and so in trampolining, in a competition, you have to do 10 skills in a row, and you have to try to stay in the middle of the trampoline, you have to try to go as high as possible, and you have to be really neat and keep your body very straight. And so I did my 10 skills. I was learning to kind of chain skills together, because in diving, you do one dive and you land in the water and you can't bounce off the water and do another flip. So I got through all my 10 skills, I stopped in place, and that was my first competition, and it was a bit of a competition of attrition. I think some other people made some mistakes because somehow I ended up winning, and I have no idea how that happened. But that was a very good start to the trampolining year. I also got up to lots of other fun social stuff, so this is kind of right at the end of Michaelmas. I experienced my first white Christmas, which is beautiful. This is just outside the window of my room. This is what Brasenose College looks like in the snow. My course mates and I enjoyed the snow for one morning and then had to be in classes for the rest of the day. Here I am with my friend Mario. I spent Christmas at my friend Anna's place in Dorset. Here we are celebrating in the hall. And I want to tell you about my beautiful friends. I got to meet some people that were not only incredibly kind, but also incredibly inspiring and did so many cool things. So, because each college has people from lots of different courses, you get to meet so many interesting people. So I'm gonna laser point to these people. So this is Zoe, she's my flatmate, and she does mathematical computer science. And we had some really interesting conversations because she did one of her research projects on computational modelling of Alzheimer's disease, modelling how it progresses through the brain. So that was really cool to kind of have that interdisciplinary intersection. That's Ethan, he studies neuroscience. This is Kate. Kate really inspires me. She's on the path to become a human rights lawyer. She's from America, and she just does part-time work for one of America's biggest whistleblowing law firms, just casually on the side. This is Matt. He does computational chemistry, and he is one of the most British people that I met in my whole year in Oxford. He taught me how to make a cup of tea. This is Julia. She does linguistics, and once she came along to a neuroscience talk with me, and at the end of the talk she put up her hand and made a complaint because she felt that the neuroscientist giving the talk hadn't used the proper definition of what a word was, because it was a neuroscience talk about how the brain processes language, which I found hilarious. This is Anna. She does health data science. She's doing a PhD, so I'll be continuing with Anna and with Mario, who does maths as well. This is Jim. Jim studies languages, and he decided that learning other languages wasn't enough. He needed to create his own language. So he has his own language. It has a whole religion associated with it. He's taken his favorite grammar rules from a whole bunch of other different languages. He also has bespoke poetry written in his own language. It's just incredible. And this is Adelaide. She does ballet and English and is combining the two in her research. So I got to meet some pretty awesome people. And then right at the end of this first term, the end of Michaelmas, my quest for the DPhil met another milestone. This was on my birthday. We made gingerbread cookies that said, "DPhil funding, please." And this is a little gingerbread scientist with a lab coat. So this was my prayer for some funding. Unfortunately, it wasn't answered. I got two offers to do my PhD at Oxford, but neither of them came with funding. So I knew that this was kind of a good first step, but the hardest part was still to go. I still needed to find a way to support myself while doing my PhD. And this is the time where it really set in the pressure of getting a distinction in my course, because I knew that if I wanted to apply the following year, that was kind of the benchmark that I wanted to meet. So that was the end of Michaelmas. And now we're on to Hilary, the second term. Still not sure why it's called Hilary. So I continued my course. I started my first research project. And in this research project, I used this technology called optogenetics. And in this project, I was using it to study the way that neural signals in the somatosensory cortex, so the part of the brain responsible for processing touch, how that part of the brain communicates with subcortical brain regions. So parts of the brain like in our brainstem or in a region of the brain called the thalamus. And that's an essential process to be able to communicate from the somatosensory cortex to the rest of our brain and to inform our actions. So it's a really fundamental mechanism that the brain needs just for normal daily operation. Oh, have we got an issue yet? Oh, I mean, we can save a seat. There's a seat there.
He can see himself in the vision.
I thought he could see me. That's okay. That's right. So I got to use optogenetics in this research project. Throughout this term, I did lots of hard work. Sometimes I worked late into the night. Sometimes into the early morning. I gave a presentation. I presented a poster on my research. But I also wanted to kind of give you guys the neuroscience scoop on the technology that I was using, optogenetics. So optogenetics is this really exciting technology that's really only become mainstream in neuroscience over the last 15 or so years. And the original discovery of optogenetics was based on some findings about green algae. So who would have thought that discovering green algae could help neuroscience? But I'll show you how. So it was found that in some of these green algae, they would grow towards light, right? And that makes sense because they need light for photosynthesis. But how they were doing this was unknown until they discovered this light-sensitive ion channel. This is a complex diagram, but the most important parts are that when blue light comes in, that allows sodium to pass through the channel. So it's a light-activated channel. And if we take this protein from algae and we make brain cells express this protein, then the neurons become light-sensitive. So we can control brain cells with light. So this is a neuron here. This is a very stylized diagram, but what it's meant to illustrate is that when light hits the neuron, it activates these light-sensitive channels, and the neuron fires, right? It sends an action potential, it sends a signal on to all of the other neurons that are listening to it. And I've got a video to show you to demonstrate kind of the power of optogenetics. So this is a use of optogenetics in a live mouse. So the mouse has a fiber optic cable implanted in its brain, and it has this light-sensitive channel being expressed in a specific region of its brain as well. And what you'll see in this video is the mouse standing normally, and then the light will come on, and we'll see what happens. Alright? So the light's not on at the moment. And then it turns on, and the mouse immediately starts running in circles. And that's because this light-sensitive protein is being expressed in the motor cortex. So when the motor cortex is activated, this mouse starts to run in circles. So this is showing that optogenetics can be used in a live animal, and this is an incredible experimental tool to figure out the function of specific brain circuits. It's also has potential therapeutic applications. So if you imagine, for example, people who have degeneration of the light-sensitive neurons in their retina. There are currently gene therapy trials being run in Australia that are seeking to replace these degenerating light-sensitive cells with cells expressing this light-sensitive protein. So optogenetic can be used for experimental applications, but the way I was using it in this project was an even more advanced version, which only exists in Oxford and a few other places. So there are lots of labs in Melbourne using optogenetics, but this special type of optogenetics called two-photon optogenetics, where 2 photons of light converge at the same place to activate this channel. It allows us to get single neuron resolution. So this is 2-photon imaging, which is a little bit different, but you can see that we can see individual neurons as they're active in an awake mouse. So in this mouse, there's a window that's been placed over a part of the skull. The skull's been removed and a window has been put in place. And so using a very specialized microscope, we can look at individual neurons and track their activity. So pretty awesome technology, and that was a really incredible experience to be able to go to Oxford and do things that I wouldn't have been able to do in Melbourne, and kind of learn some of that unique knowledge that I might one day be able to bring back here. So that's the segue into optogenetics. I'll go back to Hillary term. I did some more trampolining, and the big part of trampolining in Hillary term was the varsity match. The varsity match is the big match between Oxford and Cambridge. It's the grudge match. So there's a bit of a rivalry, and in the trampolining version, I was one of the final competitors to go. It was looking very close, and so I knew that a good routine would set us up really well. So all the pressure was on. So this was my routine. Since the last routine that you saw, I'm doing more flips, I'm going higher, I'm hopefully keeping it neater. I started off with a front double with a half twist. There we go.
Oh.
And then the rest of and then the rest of the routine, I'm just trying to keep it clean, stay in the middle. Oh, I moved a bit. Alright, and the final skill I'm finishing off with another double.
Oh.
There we go. that was the routine. And so, this was a triply successful day because I ended up as varsity champion. Oxford won against Cambridge, and I also ended up with a full blue award, which is the award that you can get from Oxford by being doing well in any different university sport, and you have to get a certain place at the varsity match. So that was a fantastic day for trampolining and a big highlight of the year overall. There was lots happening just in general life, but it was winter. So I took photos of the sun while I could get it, but it was raining a lot. Most of the time we were inside. I introduced my British friends to Weet-Bix. Did lots of fun college traditions. Here we stood on the tables in the dining hall and sung lots of songs. I went to my other friends' colleges and got to explore lots more of Oxford that way. The quest for the DPhil was still ongoing. And in Hillary term, nothing happened. I was just waiting. I was just waiting and waiting. I was applying for lots of external scholarships, but not hearing anything back. And so this was kind of the my anxious wait for figuring out where I would be next year. Would I be coming back to Melbourne for my PhD? Would I be staying in Oxford or going somewhere else completely? So I was a bit in limbo there. But that is the end of Hillary term. So we're coming on to the final term, Trinity term. And this was the most intense term of the three. I took on my second research project. And in this research project, we were trying to figure out a way to get neurons which are derived from stem cells to connect with each other faster and more strongly. So we were trying to express this special protein that made neurons more likely to wire together during their early developmental stage. And I worked really, really hard on this. I spent lots and lots of long days in the lab. I saw lots of beautiful neurons. I recorded from individual neurons using this technique called a patch clamp, which I talked about in another class way back. And worked really hard on this project, but at the end of all of it, ended up with a negative result, which is scientific speak for the hypothesis was wrong. So that was really heartbreaking, because the hypothesis was really interesting and would have been really cool if it worked out, but it is also just a part of science that sometimes it doesn't. So I tried to make sure I did a really rigorous job of proving that we were completely wrong, and ended up getting to the end of that term and submitting this thesis, which I had worked really hard on and thought I'd done a really good job, but hadn't quite shown what we thought it would. There was lots else going on, and in Trinity term, it was summer, which meant we could actually go outside. Here we are punting on the river in Oxford. We're spending lots of time playing games outside, eating outside, fantastic. Here I am floating in a bubble I'm not sure if you can see that, floating in a bubble in my college. And yeah, lots of Oxford traditions. There was lots of fun stuff going on outside the studies that made it a great year. And we ended the year off by going to this awards night, which was the Oxford University Sport Awards for all of the sports people in Oxford coming together. I was there with my beautiful team and my beautiful friends, and ended up coming away with an award. I got for the best first year student, and got to celebrate that with everyone, which was very special. It was also a big moment in my quest for the DPhil. So in Trinity term, I heard back about one of my applications. Not from Oxford, but from Cambridge. So Cambridge gave me an offer to do my PhD, which I was super excited about, and crucially, they offered me funding. Oh. So they were willing to give me a scholarship for however long it would take me to do my PhD. And I thought that was super exciting. So I took that offer letter and I said to Oxford, you know what? If you don't give me funding, I'm going to go to Cambridge. And the rivalry runs so deep that somehow, miraculously, Oxford came up with the money. So Oxford then counter-offered me to give me a scholarship to do my PhD there. And it was a very hard decision. I went to Cambridge with Kate to check out the city and visit it. But ultimately, I decided that the experience that I'd had with my lab in Honorary term, the first project where I used optogenetics, that environment was where I thought I would succeed in a PhD. So I decided that I'll be staying in Oxford to do my PhD, and I'll actually start that in October, which I'm incredibly excited about. So that was kind of the quest for the DPhil complete. There were still some final big milestones to go in my course. I still had to submit my final thesis, and this was the most intense part of my life, the most academically stretched I've ever been. So I submitted my thesis at about 3 a.m, sitting on the steps in the same square that I'd walked into when I first came to Oxford 1 year later, and it was a great moment to reflect on all of the things that I learned, the really tough times, the really great times, all coming full circle and being able to celebrate with all of my course mates and I who are now finished. So that was the year in Oxford, Michaelmas, Hillary and Trinity, all coming to an end, except not quite, because there was a little bit still to go. There's an epilogue to this. So I came home after a year of not seeing my parents, a year of not being in Melbourne. I was trying to sneak up on them and surprise them. But we still had one milestone to go, which was the viva, which is an oral defense of your thesis. So I had to defend my thesis against 5 Oxford professors. Here I am surprising them. So we still had the viva, which even though I did it from Melbourne over a video call, I still had to dress up in the fancy academic dress and talk to, it was actually 4 Oxford professors, and 2 of them are the kind of lead examiners, and just my luck, I had the 2 heads of department that had read my thesis and were going to question me. There was also a 5th examiner who was the external examiner, so they can't be from Oxford. And also, luckily enough for me, it was a professor from Cambridge, but not only from Cambridge, it was one of the professors who I had an offer to do my PhD with and had rejected. So I was mortified, but it all seemed to go well enough. And to my surprise, 2 hours after I finished this viva, we got our final results back. And so I was very, very nervous because I had just been absolutely incredibly nervous talking to the 2 heads of department and this professor from Cambridge. But I thought I had managed to keep my nerves under control and get through it okay. And we got our results back, and I was really hoping that even though I had the PhD all secured now with the funding, I was still really hoping that I could get that distinction that I'd been aiming for all year. And not only did I get the distinction, I got a prize for being the dux of my master's cohort, and it was called the Sherrington Prize, which I don't know if you remember way back when I first came into this building. It was called the Sherrington Building, and Sherrington was the person that mentored Howard Florey, which the Florey Institute was named after, and where I did some of my research in Melbourne. So that was very, very special, and to know that, you know, all of your support in me, all of Rotary's support in me, who funded me and allowed me to do this, had motivated me to work really hard to get to that point, was really, really exciting. So that kind of brought the Masters to a close. So I guess the question is, what's next? Well, at the moment, I've been back here in Melbourne, spending lots of time with my grandma and my family, and just trying to relax as much as possible before I start my PhD. And when I go back in 2 weeks' time and start the PhD, I'll be with my 2 PhD buddies. These guys are in the same lab as me and starting at the same time. And I'll be using this technique called optogenetics. I'll be using the special 2-photon optogenetics to target single neurons. These are slices through the cortex of a mouse brain. And I'll be investigating the way that activity in the visual cortex relates to the perception of our visual environment. So I want to understand how the activity of neurons in the visual cortex determines what we see. And I'll be using mice for my research, because when we use mice, it allows us to have this incredible resolution and look at individual neurons. So I'm really, really excited for that, to be able to use technology that doesn't even exist in Australia to be able to do this research. And I wanted to kind of give some sense of what my research might be building towards. This isn't work that I'm going to be able to do in the next 3 years, but eventually the idea is that if we better understand how the visual cortex encodes visual information, we're better able to restore vision when it's lost. So the way that would be done is through brain-computer interfaces, so having a device that stimulates neurons in the pattern that's required to perceive the visual environment. And that hasn't been done yet, right? The groundwork still needs to be done. But there are brain-computer interfaces that are in use now in trials and even in healthy participants. And I wanted to show you a really cool video of a brain-computer interface that's working now. And this is being developed by a neurologist who did his training in Melbourne. He's now in California, in Silicon Valley. But he did his training in Melbourne, and to his middle cerebral artery, and it's just like a stent, so it presses up against the outside of the blood vessel, except different from a stent, it has electrodes on it, so it can read brain signals. And what it does with those brain signals is it passes them into a device. So this patient has an iPad, and using this device, they're able to think about what they want to type to their doctor. And based on that, this is a collaboration with ChatGPT. So ChatGPT generates plausible responses that this person might want to send to their doctor. And using this device, they're able to select the message that they want to send. And that way they're able to communicate with their doctor without moving their limbs. So that video has been really inspirational for me in kind of showing what's possible in that space. So hopefully we'll get to watch it later. But that kind of brings the epilogue to an end, brings the best year of my life so far to an end. And the last thing that's left to do is for me to say thank you so much. This class allowed me to work on my presentation skills. It let me explore a whole bunch of neuroscience topics that I was really interested in but maybe didn't have time to look at in my classes. And most of all, it was awesome just to have your encouragement and belief behind me while I was in Oxford this year, and to read your lovely messages. So thank you so much. Thank you for having me back. And I'd love to chat about any questions that you might have. So thank you.
Now, I may have misunderstood what you were saying about the light, and is that going to apply to people, say, who were born with no sight, as against people that have lost their sight?
Yeah, so it'd be much more easily applied to people who are in early development to fine-tune those networks to have the sight that most of us enjoy, and so that's a much more difficult challenge. So the things that we're aiming to look at first are acute loss of vision, so later in life.
You use genetics in the genesis part of it or in the nature part of it?
So yeah, so the reason it's called optogenetics, so opto meaning light, but genetics is because this protein, this channel that I showed you that lets ions through and activates the neuron when light shines on it, it's genetically encoded in the neurons. So there are lots of ways to do that, but the way that we do it is using a specialized virus. And the virus goes up to a neuron in a dish, and it puts the genetic sequence for this channel into the neuron, and then the neuron starts making it itself. So that's why it's called optogenetics.
How long do you anticipate the DPhil to take, and what are your plans after that?
So the DPhil takes 3 years on paper. Most people take 3.5 to 4. I'm aiming to do it in 3. So that'll be how long I'm definitely in Oxford. And then after that, there are a lot of different possibilities. I want to stay in science. I want to do scientific research. That's what I've wanted to do for a very, very long time. And so that's still my goal. In terms of where I'll be, Compared to 1 year ago now, I mean, today is the 17th, so 1 year ago today it would have been my third day in Oxford. The world feels a lot more open to me now than it did a year ago. Before I went to Oxford, my world was just Melbourne. And now having friends that I've met this year who have now gone home to a whole bunch of different places around the world, and learning about the cutting edge research that's being done in all of these places, it definitely makes me think about all of the different possibilities. And I'd still love to come back to Melbourne and do my work here. But I think one of the things that I love about science is that it's not nationalistic, or at least neuroscience. I hope that even if I'm not in Australia, the work that I do and the contributions that I make to the scientific community will still help people in Australia, in Melbourne, and in my local community, even if I'm doing it from afar. So that's the plan. Fantastic. Shall we have some morning tea?
Yeah.
Thank you, guys.
He's our honorary grandson and will be with him on his journey no matter where he goes. In our hearts.
Oh.
Yeah.